All right. So we're ready to start the last talk of the day. So Tang from Shanghai Jiao Tong University is going to talk about wall boundary conditions for self-propelled particles. Thank you.
Thank you. Actually, I changed my topic because I think this one fits better the workshop, because the first one I put is too applied, and then I realized there are a lot of analysts here in the audience. So it's better to talk about something that maybe there is some interesting analytical problem here. So what I'm going to talk about is about the boundary conditions for self-propelled particles. So one part of my work basically is for chemotaxis. If we consider about chemotaxis, it's a multi-scale phenomena. People can start from what happened inside the cell, how the cell sense the environment, to how the cell would move. This is a typical trajectory for the bacteria to move. It would just go straight forward, stop somewhere, and change its movement direction, then go to another direction again. This is at individual level, where at the population level, usually people use the density to describe the density evolution for the bacteria. So the time scale for this whole process actually change a lot. What I'm going to talk about today, basically, it's for the scale from here to here. So if we look at the scale here, actually it's kind of already very standard in the literature for chemotaxis. So if we look at the trajectory of the cell, then people are going to use the kinetic equation to describe it. Well, the F would depend on x, space x, and the time t, and the movement direction v. Well, the how it senses the environment would be encoded in this tumbling kernel. This is called the tumbling kernel. It would depend. This S is the outside signal. Then how much it would want to change its movement direction would be determined by the outside signal or the gradient of the signal. Then actually, if we look at the literature of classical neutral transport equation, this kind of equation is already studied a lot in the community for neutral transport equation. Then if we look at the literature for people studying neutral transport equation, there are two different kinds of limits. The one is called the Fokker-Planck limit, which basically describes the thing that when the particle moves forward, it would change its movement direction a little bit in each of the scattering, because it would change its movement direction all the time, but then depend on how much it change for each of the tumbling. Then this is a phenomena for the Fokker-Planck equation. Basically, it means that it changes a lot, and in each of these, it tumble a lot, but in each of the tumble, it would change its movement direction only a little bit. So this is a highly forward peak case. Then what you're gonna to put in your tumbling kernel, you would put something like this kind of stiff term here. This term basically encoded the fact that each of the tumble, you would just change your movement direction a little bit. Well, the frequency here, this 1 over epsilon to the power 2 here, it means that it needs to change a lot. I mean, the frequency it changes is a lot, but each of the time it just changes a little bit. Then when epsilon goes to 0, what people are going to get is this Fokker-Planck limit. Well, there is another very popular limit, is diffusion limit, which is sometimes we also call it parabolic scaling. Then what people would put is that it tumble a lot, but in each of the tumble, each of the movement direction change, it would just when it changes movement direction, it's just that they find a new direction randomly. So it's kind of isotropic scattering. Well, this one sometimes called it's highly forward peak scattering. Well, for these two different cases, both of them, the movement direction of the cell change a lot, but it's only depend on how much it's changed each of the time. It would give you two different kind of limits. for this diffusion limit, when epsilon goes to 0 here, then what you're going to get for the community of neutral transport equation, what you're going to get is just the diffusion equation. But if we consider about the classical case, which means that in your atomic kernel, it would depend on your S, outside signal S or gradient of S, then you're going to have additional drift term. This is kind of already very classical case if people want to consider about the limit for the for chemotaxis or kinetic model. But then what we want interested in this talk is about what going to happen at the boundary. Because when cells at the boundary, then there will be a wall. Then actually people already, if you look at the literature for people to do fluid self-propelled particle simulations, it's already very well known that when the cell, it would have a head, it would have a flagella, and the flagella would push back the water. So actually, they would form a dipole inside the fluid, and in the direction of its head, the fluid would just go forward, and in the direction of its flagella, the fluid would go backward. Well, for the other part, the fluid would flow like this. Then this is why there will be some flow go down here and from here to go up here. So this is called basically called the dipole. You can imagine when it's near the wall, because of this fluid dipole here, then its effect would push the cells to the wall, basically. So so this is the phenomena people already observed. And the other phenomena is that you can see that even if without this kind of fluid effect, because the cell head is, you can consider as rigid body, because the cell have its self-propelled particle. When it go to a wall, they still have a flagella to push it to the wall. They don't want to change its movement direction immediately. Then they will spend some time on the surface of the wall, which is quite different from the classical particle, because when classical particle collide to the wall, it will just bump back immediately. But for the self-propelled particle, the phenomenon is quite different. You can see that they would stay on the wall for some time, and either because of the fluid thing, or just because of the self-propelled force by their flagella.
Right, what you're describing physically is the border taxis, right? It's called behavior.
It's not.
It's like slips to the border a little bit, border taxis.
And I don't know that for this actually there's no word about taxis, right? But just when it goes to the wall, they want to stay near the wall. Right. But the physicists would debate the hydrodynamic is more important, or the pulling of its flagella is more important. This is the argument they are trying to argue. Then actually, if we look at the population level, this is what will happen, because this is one experiment in the literature. So there are two plates. Between these two plates is a droplet that full of bacteria. Then they just measure the density distribution between these two plates. This is what their observation. Then they will see that the density near the wall would be much higher than between the wall. This y-axis here is the magnitude here. It's the two plates are very close to each other. Then you can see that the density is much higher near the wall, but in between it's much lower. Well, then some physicists write down a stochastic model out of it. Then you can see it's kind of very simple. The movement velocity of the bacteria is v. Well, you just project it to, it's just considered as a 2D movement. Then this v is just its movement it's the velocity of its movement, while this sine theta t is just a project to the direction of this two plate, which is the direction here. Then when it's inside the wall, or when it's at the wall, but its movement direction is towards the wall. The main we can see it's more clearly from. Oh, okay, I will explain it later. But anyway, if it's stay at the wall and its movement direction is toward the wall, then it won't change its y, the position. But if it's at the wall, but its movement direction is outward, then they can move according to the law here. This is a very simple model where its movement direction is just by rotational Brownian motion. Then by using this very simple model, they can recover kind of the experiment here. They can see that there will be more bacteria at the boundary, while less in the middle. Then what I'm going to do is to basically, this is a full part of my talk. The first part, I'm going to write down a velocity jump model. When I mean velocity jump model, basically the kinetic version of the, I already mentioned just now what kind of boundary condition. If we write down a kinetic equation, then we consider about that all the bacteria are between two plates. Then what kind of boundary condition you should put for that kinetic equation. This is the first part. Then afterwards, we can derive these two different asymptotic limits out of the velocity jump model. We can find out what kind of boundary condition you should put if we consider if the cells are as either for the Fokker-Planck regime or as a classical regime. Then we can give you the boundary condition what it look like. So first of all, the velocity jump model. Actually, if we look at some of the experiments, they already know that the tumble, the movement, this t, the parameter t I put there, the t would decrease if you are near a surface. This is what they already observed in experiment. The surface, near the surface, the tumbles are suppressed. Well, of course, for different bacteria, how much it would suppress is different, and the movement direction would be just along the plate. This is the physical observation. Well, if we write down a model, actually, it's kind of just use the simple model that already proposed by physicists. It's like inside these two plates, the cells would just do. For this one, right here in the middle, it's just to run and tumble. Well, when it reaches the boundary, then it still can tumble, but when its movement direction is in towards the wall, then it cannot leave. But if when it changes its movement direction until its movement direction go outward, then it's just to leave the wall. So this is a very simple idea. Then when we write it to couple this to the kinetic equation, what you're going to get is like this is the inside the wall, inside the domain, then which is the same as the kinetic model, classical kinetic model. What difference is these two equations? This n plus is the number of cells that attach to the upper bound, while n minus is those cells that attach to the lower bound. Well, you can see that even if the cells are attached to the boundary, they can still change its movement direction according to some tumbling kernel. Well, there will be something. This is a v sine theta, and actually it's kind of like the contribution of those cells that move to the boundary, because this n is those cells inside the domain. Then those cells, when it's reached the boundary, they would change into cells that attach to the boundary. This is why where the source term come from. Well, what's interesting is basically the boundary condition. If we think about the boundary condition for theta is the movement direction, then it will satisfy the periodic boundary condition, of course, for n. Well, then the thing is this interesting boundary condition here. It's like for those cells attached to the upper boundary, they would that move with direction theta prime, they would change its movement direction by tumble to theta, but then it's gonna to change to the theta direction. Those theta, if the theta direction is outward, so because this one is for the upper bound. So we have an upper bound, which is the n plus, and here is the n minus. So if the movement direction of those upper bound cells, when it changed to minus sign, the theta becomes from minus pi to 0, then it can leave the boundary, then it would contribute to those change to those cells that's inside the boundary. Well, if it's at the lower bound, then when its movement direction changed to 0 pi, then it will go to cells inside the boundary. So this is the two boundary conditions we put for n plus and n minus. Then we can check there is conservation of mass for this three equation system. And this is kind of classical. Then what kind of theoretical result we can get? We can get the relative entropy inequality for this. Basically, this relative entropy inequality would give you the fact that the non-time convergence of this system. But you need to, the difficulty for this relative entropy condition is that we need to combine all these three equations together to give you an inequality. Well, and after we get the relative entropy as inequality, we can get the long-time convergence. But what I want to emphasize here in this talk is the convergence to the Fokker-Planck model. Basically, when it goes to the limit of this Fokker-Planck limit, what kind of boundary condition would what the boundary condition would be like afterwards. But here, from this picture, we can kind of like understand what happened for the let me see, for the system we have. It's like initially all cells are in the middle. Then after some time, some of the cells that here is minus from minus pi to 0, those cells that can go to the wall. But for those cells that go outward, they would form some kind of vacuum here because they just go outside. Okay, so then we look at what kind of boundary condition we should put for the Fokker-Planck equation. Then if we start from the model here, because if we think about the Fokker-Planck equation, then actually we can look at what kind of if we think about it at the individual level, then what's going to change here actually is just changes how the cell changes movement direction to the Brownian motion, rotational Brownian motion. Then what you're going to write down would be a Fokker-Planck equation in your theta direction instead of this velocity jump process. This is the Fokker-Planck equation. Let me. I start from there. I think it's better to explain here, because as I already mentioned that, because if we want to get the Fokker-Planck limit, this is the scaling we put in the classical kinetic equation. Then we're going to have this theta minus theta prime divided by epsilon, which means that in each of the scattering, we only change a little bit the movement direction, and we need to put 1 over epsilon to the power 2 here in order to mimic the fact that it changes movement direction a lot. After put this scaling, when epsilon goes to 0, we're going to recover from the velocity jump model to the rotational diffusion here. Then because at the boundary, it's the same here. It is at the boundary, what we're going to have is that the cells would the way that the cells change its movement direction would be the same as its inside the boundary. So these scale, after you put this scaling, what you're going to get still the rotational diffusion in theta. Then this is just a typical example for the tumbling kernel here. It's like this. It just mimics the fact that the movement direction of each of the tumble is just change a little bit, because if you look at the sign here, it's very long range. But because that variable is the theta divided by epsilon, is the singularity here. So this is what happened in the middle. But what's interesting is what happened at the boundary. What happened at the boundary is here. It's like before, for the velocity jump model, the boundary condition is to, after we put the scaling for this kind of small change in movement direction and the frequent velocity, frequent tumbling, then what the scaling gonna put here would become like this. Well, these are the two scaling we put for the velocity jump model. Then for the first periodic boundary condition in theta, it would become the same periodic boundary in the limit. It would become the periodic boundary condition for the diffusion in theta, which is these two equations here. But well, for the system here, it would change into three equations. I will explain each of these three, what's the meaning of these three equations later in previous slides. Well, let me look at here. Okay, so actually this kind of population level Fokker-Planck model, as I already explained, in the middle, it would just be the theta movement, rotational diffusion equation in theta. Well, for the theta at the boundary, the periodic boundary in theta is here. Well, because as we already explained, because for the kinetic equation, each of the change of movement change, it's just a change a little bit to your velocity direction. So for those particles, leave the plate. It would just leave the plate immediately as far as its movement direction changed outward. So it won't be any particle that go in the movement direction to, for example, for the particle on the upper bound, when it would change from n plus to n, but because each of the time it's just changes movement direction a little bit, so all of those particles leave the plate. As far as it change to the movement direction outward, it would immediately leave the boundary. This is why it would give you that. Actually, for those, this quantity gives you the fact that those cells at the boundary, near the boundary, it's just some place here, near the boundary, but very close to the boundary, but its movement direction, if its movement direction go outward, it would go away immediately. So this is why it should be 0 here. Well, for those, this condition would tell you that for those, because those outward going particles, when those particles that attach to the boundary, as far as this movement direction reach 0 and pi, it would switch to inside particles immediately. And this is why at the two boundaries, this 0 and pi here, the condition should be 0. It can, when the particle attached to the boundary, their movement, as far as their movement direction is along the boundary, they would immediately change to the p type. Well, so we have already two boundary conditions, one for p and one for p plus. Well, we still need the boundary condition, one additional boundary condition for the flux, basically. There are two conditions here. These two conditions basically tells you that for those flux, for those cells that attach to the boundary, because they would do the rotational diffusion to leave the boundary, then this rotational diffusion would provide you some flux for this P plus here, And this would provide you the flux inside the domain. And this is why actually the flux inside the domain at the place where theta equal to 0, there will be some particle switch from those attached to the boundary to those inside the boundary. This is why there will be a jump when theta equal to 0 for those cells inside the boundary. Well, the jump actually is just for those coming from the flux of those cells that attach to the boundary. So this is the specific boundary condition we need to put for the Fokker-Planck equation. Well, this kind of condition, we can derive it from, also for the Fokker-Planck equation, we can check that there is conservation of mass. You just sum up, take the integration and sum up all those three equations. You can get the conservation of mass. Well, we can look at the numerical results. What happened there? The numerical results basically are what I present here is P plus, P minus is for those number of cells that attach to the boundary. Initially, we start from all cells that are just inside the two plate. Well, as time goes on, you can see that all this there will be accumulation of cells at these two boundaries. Well, if we look at this P, this number here basically is for those cells that near the boundary, but they're inside the domain, but they're not attached to the cell, which is the cells here. It's like they're not attached to the domain, they're already inside the domain, but they're very, very close to the boundary. This is for those cells there. So you can see that those cells initially, because inside the domain there are cells, but you can see there will be some flux coming from those attached to the boundary. The reason why there will be a peak here is coming from the cells that attach to the boundary. And you can see that there will be a jump in the derivative of your theta, because this is a theta distribution. So you can see that at the 0 point and the pi point, there will be a discontinuity in your theta derivative. And this coming from the flux that generated by those cells that attach to the boundary. When they switch to those cells inside the boundary, they would generate this kind of jump in your flux. Okay, so then we kind of like understand why this kind of one integral kind of boundary condition would generate these three. Actually, it's two, three boundary conditions for your Fokker-Planck equation. Because first of all, because when you for those cells that go outward, when it's very close to the boundary, there cannot be cells that for those cells very close to the boundary, they have to leave. They cannot near the boundary. This is basically what this is due to the fact that each of the tumbling, you only change a little bit your movement direction. While the other two coming from the flux. And this flux, you can derive it also, you can understand it also from the scaling here, because each of the time you only change a little bit of the direction, then all those cells can only exit. If you think about the exit movement direction of the cells, they can only exit at the 0 point or the pi point, because each of the tumble, they just change a little bit of the movement direction. And for those cells that attach to the boundary, they can only take value in the theta the theta variable can only be 0 to pi, because you can only consider those cells move towards the boundary. Only those cells can attach to the boundary. So the definition domain for your n plus would be between 0 to pi if you think about the theta variable, and because each of the time this n plus would change a little bit its movement direction when it's switched to those cells inside the domain. They can only appear at the place where theta they can only appear as a switch to the cells inside the boundary at the place where theta equal to 0 or at pi. So you can kind of like understand this kind of limit by the physical intuition. But of course, we make this kind of proof rigorous by just multiply by some test function, think about the weak convergence, all those kind of techniques. Then if we look at the numerical simulations, here is the same. It's for those. This is for those cells that attach to the boundary. Well, those are the cells that are very close to the boundary, but it's already inside the boundary. So you can see there are always peak here. Well, for different epsilon, then the peak become more and more obvious, and you can see there will be distribution for the cells inside the cell. Then the boundary condition for the Fokker-Planck equation, the theta direction here for those attached to the boundary should be 0 on the two sides. And when epsilon is not very close to 0, then there will be a jump here. Okay. Now, actually, we can extend this kind of derivation and we call it confined Fokker-Planck model to other geometry, which is it's more complex, but I can try to explain the relation with the physical experiment, because in physical experiment, actually they really put some cells inside the domain and let it go move inside the domain, and then trace the trajectory of the cells. Then they just measure how many of the cells are attached to the boundary and how many of the cells are inside the boundary. Then this is their observation. Their observation is that they just make their disk, just let the cells run in the disk. Then they increase the size of the disk and measure how many of them are attached to the boundary and how many of them are inside the domain. And the result is like this. As far as you increase the size of your disk, then there will be less and less cells attached to the boundary, and there will be more cells in the middle. Then they just do experiment trying to plot the curve, like if your curvature change, if your plate become bigger and bigger, how many of them would the number of the cells attached to the boundary, how it's going to decrease. This is an experiment. Actually, there are also other geometries which is more interesting. It's like they just make different geometry. Look at how the boundary accumulation affected by different geometry. You can imagine if the curvature is for those curvature bigger here, then you can see more cells here. And actually this kind of phenomena, we can recover it by our model. Then if we only look at the individual basin, and we think about how the cells move when it's inside the domain, or that actually we put two cases. The first case is that the cells are moved inside the disk. Well, for this case here is kind of like cells are moved outside here. Then what we need to consider is about how the cells would interact with the wall here. Then you can imagine that if the cells attach to the go to the boundary, then if its movement direction is tangent to the movement direction, it would just and the cells, not only it have a position, but also it have a movement direction. If its movement direction is towards this direction, they would kind of like attach to the boundary, but it does not mean that they don't move. Because if the movement direction is like this, then along the disk, it can still sliding. Even they attach to the boundary, but they are sliding along the boundary. But if the cells are moved outward here, then when it go to the boundary, and if its movement direction is like this, it would slide like this until its movement direction is like this, and then they escape. So you can see that the way how they interact with the wall is different for these two cases. Then if we want to describe this, of course the model is more complex, but we can do it. I just trying to tell you the variable we need to use if we want to model this kind of thing. Then we need to put, for example, when we describe the position of the cell, we need to use the R and the C. Basically, this R is the radius position, while C is where it stay. The angular variable where it stay. Well, its movement direction would be described by theta t here. And one can write down a similar model in similar principle, but more complex geometry like this. And its movement direction, so you can see that its movement direction is still changed by rotational diffusion, while the C is how its position going to change, because it's just you are in a disk, then you just need to think about I think the important thing here is like the velocity, how you project your velocity is more important here. It's like it's not only relates to its movement direction, but it also relates to where it stays. So actually what matters is the difference between these two angles would determine how it changes direction in this R variable. Okay, so I'm not going to trying to explain more details about the process here, but believe me, when it's the velocity in your R variable would be V cosine theta minus C, because C describes the position, while theta describes its movement direction. And basically, so you can see that if, for example, if we think about this red cell here, and its movement direction is this red angle here, this red arrow. Right, this red arrow here, then this C minus theta gives you this blue, what I have blue here. Then you project it to the R variable would give you this variable. Well, if you project it to the C variable, would give you sine theta minus C. So this P and Q, and P is the cells inside the disk, while Q is those attached to the disk. You can still write down a model. It's like in the theta direction, it's still rotational diffusion. Well, for the thing change here is about the movement, how its movement depends on your theta and C. Well, this is the model. If we consider about a disk like this, then we can do similar simulation. So what's the details of how we write down all those kind of boundary conditions, I won't discuss it because there are too many details here, but the principle is the same. It's about you need to consider about those outward ones. Outward ones would leave the boundary immediately. Well, we will have periodicity in your C and the theta variable. All those kind of things we can write down all the details of the boundary conditions for the Fokker-Planck equation. We can even consider about the annulus. It's like for the cells move inside the annulus, there will be an obstacle in the middle where cells are moved here. Then we can write down the boundary conditions for these two boundary and consider about the population level distribution. Well, the phenomena, if we look at the phenomena, actually it's similar. So you can see that these are the numerical results. You can see that if for those near the inside the boundary, at the boundary, then the number of cells would increase, become more and more. And here is those cells that's inside the boundary, but near, but it's not attached to the boundary, but very close to the boundary. Then what you're going to have is then you increase your size. For example, you can increase the size of your domain. You can see very similar phenomena as the experimental result. Well, you can put the cells in the middle of the annulus. You can measure how much of the cells would like to attach to the boundary here, and how much of them would like to attach to the boundary here, and then you can just compare the number of them and measure how it's going to change. For example, the diffusion coefficient. The bigger the diffusion coefficient is, then the more cells would inside the domain. But the smaller the diffusion coefficient is, then there will be more cells attached to the boundary. All those kind of things we can explain this by this model. So finally, I will try to. Actually, all different models. All of them would start from the velocity jump model, but you take different limits. Think about different geometries. You can always get very different model. Well, but if we think about the classical limit, the story is kind of different, because for classical limit, if we think about the classical particle, when the cells are near the boundary, if the cells want to change its movement direction a lot, well, the change is isotropic. Then there will be no cells attached to the boundary, because we know for the at least we know for the gas, the boundary condition you're going to get is just the Newman boundary condition. If you want your cell change your movement direction a lot, and they won't attach. But actually, what's reality happen is when cells near the boundary, they don't tumble that much. And especially this would relate to some biofilm formation. Because when cells attach to the boundary, they will generate some glue, and the glue would accumulate the cells near the boundary, and then it's very hard for them to escape in a lot of cases, which means that the tumbling frequency for cells is reduced a lot for those attached to the boundary. But when we derive the previous Fokker-Planck equation, we just think about when the cells go to the boundary, as far as their movement direction go outward, they would leave. But actually, there should be a escaping probability. Even if their movement direction go outward, it does not mean that they can really go away. So after we take this into account, we just add this kind of effect into our model. Then you can derive some very interesting boundary condition for the classical equation, because for the classical equation, what people usually use, if we think about look at the literature of those doing analytical paper, most of the time it's Newman boundary condition. But what we have in mind, if you what's in real experiments, usually it's the experiment is done by between two plates. Well, for example, this is very typical experimental setup. There will be just they just put cells between two parallel plates, and the plate is very close to each other, and they look at from upper part to look at the density distribution of the cells. But then there will be a question as what was the effect of these two plates? Actually, they know there will be accumulation of cells near the boundary, but they just ignore it. So actually, there will be a. If you really want to compare with the experiment, you need to know what kind of boundary condition you should put for your classical equation between. We look at the between two plates, it's like this is the geometry we consider. It's like here is each of the line represent one plate, and they are not so far from each other. Where cells is in between. Well, the color here is a typical outside signal field. Then for a classical equation, we know that the cells would like to go to the place where there will be more chemical concentration. Then this is realized by putting such kind of scaling in your tumbling kernel, because the tumbling kernel would be affected by the gradient of S here. Well, for the classical equation, we also know that the parabolic scaling should be used in your system. Then we can put this thing into the velocity jump model and trying to derive what kind of boundary condition we can get out of it. Then the result we can get is like this. Actually, finally, we get some. We call it confined classical equation. So it's inside the domain, it's just the same as the classical equation, but at the two plate, you can see the boundary condition is quite different. The S, there will be no chemotactic drift for those cells attached to the boundary. There will be only derivative with respect to y. The y direction is this direction here. It's a direction here. This is the y direction, and here is the x direction. And if we generate some chemical domain that have a gradient in your x direction, but this won't affect those cells near the attached to the boundary. But you can see that these two equations actually the real the condition is not complete yet. We still need two conditions. Here, these two conditions, it tells you the ratio between those cells inside the domain and attached to the boundary. So you can see that this is a very unclassical boundary condition in your y direction. It's formed by there will be switch between rho and rho plus and rho minus, and there will be a ratio between these two to density. At least I don't know other model describe the. I don't know. I mean, after we put this kind of boundary condition, how would it affect the blow up, or how it would even? I don't know much equation look like this, and this is why I think it's kind of like, interesting. So that's it. This is the of the talk. So I just make the conclusion. We just consider about the kinetic equation, and then try confine in the environment, then derive the Fokker-Planck limit, the classical limit, and we hope to look at other geometries and compare with the experiment. And here is the reference. Okay, thank you.
You have questions?